Imagine this, a simple beat or rhythm, something as basic as a ticking clock or the steady thrum of rain, could be quietly reshaping how your brain operates. It's not just about hearing the sound. It's about how that sound rewires the very fabric of your brain's network. This is the groundbreaking revelation from a recent study published in Advanced Science. What's even more fascinating is how researchers achieved this insight using an innovative tool called FREQ-NESS, which peels back the layers of the brain's intricate wiring to reveal its hidden dynamics. But here's where it gets controversial. Does rhythm truly have the power to influence the brain so profoundly? Or could it be that our brains are simply hardwired to respond to patterns in ways we're only beginning to understand? The study offers compelling evidence for the former, but it also raises questions worth exploring further. At its core, cognitive neuroscience seeks to decode how the brain functions as a dynamic system, constantly processing streams of information from the world around us. However, one of the biggest hurdles has been untangling how different brain networks, each operating at unique frequencies and spatial locations, work simultaneously. For example, when you listen to music or hear someone speak, multiple processes overlap in both time and space, making it incredibly challenging to isolate individual networks using traditional methods. Previous research often focused narrowly on specific brain regions or broad frequency bands like alpha, associated with relaxation, or gamma, linked to heightened awareness. While these studies provided valuable insights, they painted an incomplete picture. Multiple brain activities can occur simultaneously, and older tools often blurred these overlapping processes or relied too heavily on predefined categories. To overcome this limitation, the authors of the new study developed FREQ-NESS, short for Frequency Resolved Network Estimation via Source Separation, a method designed to capture frequency-specific brain networks with unprecedented spatial and temporal precision. We wanted a transparent way to see how multiple brain networks operate at specific frequencies at the same time, explains Mattia Rosso, a postdoctoral researcher at the Center for Music in the Brain at Aarhus University and lead author of the study. Existing tools either predefine regions or blur overlapping processes. So we built FREQ-NESS as a pipeline to estimate frequency-resolved networks directly at the level of the brain's anatomical sources, enabling deeper analysis of their interactions. To test their approach, the researchers recruited 29 participants, most of whom were non-musicians. Using magnetoencephalography, MEG, a technique known for its high temporal resolution, they recorded the participants' brain activity during 2 5-minute sessions. In one session, participants rested while watching a silent movie. In the other, they passively listened to rhythmic tones played at a steady rate of 2.4 Hz, about 2 beats per second. These tones served as the auditory stimulus. The MEG data were then reconstructed to estimate activity across over 3,500 brain voxels, tiny units representing fine-grained locations throughout the brain. By applying generalized eigendecomposition, GED, the team isolated narrowband, frequency-specific activity from broader background signals. This allowed them to analyze 86 distinct frequencies, ranging from 0.2 Hz to nearly 100 Hz. Each network was characterized by its prominence, how much variance it explained, and its spatial activation pattern. During the resting state, the brain exhibited a typical 1F pattern, dominated by low-frequency activity. Familiar networks emerged, such as the default mode network, DMN, active during introspection, and the parieto-occipital alpha network, peaking around 10.9 Hz. A motor-related beta network centered around 22.9 Hz was also present. These findings align with prior research showing that different brain areas tend to favor specific frequency bands. When participants listened to the rhythmic tones, however, the brain's network landscape underwent significant changes in three key ways. 1. Emergence of new networks. Auditory stimulation triggered the formation of new networks, finely tuned to the 2.4 Hz stimulation frequency, and its harmonic at 4.8 Hz. These networks were concentrated in the auditory cortex, including Heschel's gyrus, and extended into medial temporal areas like the hippocampus and insula, regions crucial for both early auditory processing and higher-level functions. 2. Reorganization of existing networks. Existing networks shifted their frequency preferences and spatial configurations. For instance, the peak alpha activity moved from 10.9 Hz to 12.1 Hz, and its spatial focus shifted from parieto-occipital regions to sensor-motor areas. This suggests that the alpha network, typically involved in attention and inhibition, reorganized itself to support movement preparation or sensory prediction in response to the rhythmic input. 3. Stability of certain networks. Not all networks changed equally. The motor-related beta network, centered around 22.9 Hz and focused in the precentral gyrus, remained largely stable across both resting and listening conditions. This stability highlights that external stimuli don't uniformly affect all brain networks. Beyond these shifts, the researchers observed enhanced coordination between slower and faster rhythms. Specifically, the phase of the auditory attuned network at 2.4 Hz modulated the amplitude of gamma band activity, fast oscillations above 60 Hz, in areas like the insula and frontal operculum. This cross-frequency coupling was stronger during listening than at rest, indicating that the brain intensifies communication across timescales to process rhythmic stimuli effectively. Interestingly, the gamma band networks weren't confined to the auditory cortex. Instead, they appeared in broader associative regions, suggesting that the brain's response to rhythm extends beyond mere sound processing. It may involve integrating sensory input with memory, attention, or predictive mechanisms. While we expected the primary auditory network to attune to the stimulus, we were surprised by the global reorganization of the entire network landscape, Rosso told SciPost. The alpha network didn't just speed up, it relocated from occipital to sensorimotor regions, hinting at a preparatory state for action. Additionally, the gamma effects weren't localized in the auditory cortex, but spanned a broader network, implying that fast activity is mediated by interactions with low-frequency auditory networks, rather than originating locally. To ensure the robustness of their findings, the researchers conducted several replication tests. They demonstrated that similar network landscapes could be detected in independent datasets, that shorter recording durations still yielded reliable patterns, and that varying sensor types had minimal impact on the results. Randomization procedures further validated their approach. Scrambling the spatial arrangement of voxels disrupted meaningful network detection, confirming that the observed patterns reflected genuine brain dynamics. In summary, even during seemingly simple listening tasks, the brain undergoes a remarkable transformation. It reconfigures itself in three main ways, Rosso summarized. First, it tunes primary sensory networks to the stimulation frequency. Second, it adjusts the frequency and spatial arrangement of intrinsic oscillations. Third, it boosts communication between slow and fast internal rhythms. In essence, rhythm doesn't just activate the auditory area, it retunes the brain's entire network landscape. Despite its strengths, the study has limitations, including a modest sample size, the absence of baseline noise data, and a relatively simplistic task design. Future work could incorporate more ecologically valid stimuli and expand the method to support broadband and multimodal applications. The brain is an extraordinarily complex system, Rosso noted. Frequency is just one piece of the puzzle. Our goal with FREQ-NESS is to provide a transparent and interpretable framework for understanding brain networks, without relying on overly complex or black box approaches. Looking ahead, the team is working on two major directions: developing a multimodal variant of the method and creating a broadband version already accepted for publication in a high-impact journal. Collaborations across European centres are underway to explore how the brain's network landscape changes under various conditions, such as altered states of consciousness, aging, or musical training. The FREQ-NESS toolbox and full pipeline are openly available on GitHub, and the paper is open access. We hope others will use it to investigate how ongoing brain rhythms shape perception and action, Rosso added. We're eager to collaborate and guide anyone interested in advancing this field. Please feel free to reach out. So what do you think? Does rhythm hold untapped potential for reshaping the brain? Could tools like FREQ-NESS unlock new frontiers in neuroscience? Share your thoughts in the comments below.